Can you see my screen? Okay, super.
Yes.
So, good afternoon, everyone. Thanks for being here to this 10th webinar. We will focus on illuminating neuronal circuits. Before starting speaking about the lineup of today, I will spend a few words describing what is this initiative. The FNIP initiative, which stands for Focus on Optical Neuroimaging and Photonics. It was born in 2021 from an idea of Dr. Anna Archetti, and since then this initiative grown, and now we have several organizers that spans different departments of the University of Padua, but also the University of Fribourg in Switzerland, and the Italian National Research Councils. Across the year, we have been fortunate also to have several supporters in various Italian institutes and also beyond Italy. The program, this initiative, revolves around three main topics. First, optical technologies, so advancement in microscopy and photonics, then the application of this technique to neuroscience, and last but not the least, a more software-oriented part that focuses on simulation and data analysis. The main format that we organize are, on one side, webinars that are held every 3 to 4 months, and they cover a specific topic of the initiative. And we organize conferences as well, FNIP days, that are held every couple of years. And during this conference, we try to cover all the three main topics. So this year, March 2018, we will host the third edition of the conference in Padua. If you are interested in it and if you want to check what we are doing and the following events, please check also the website. So currently, more than 40 international speakers from around the world joined this initiative. And across the events that we organize, we reach more than 500 attendees. Now, moving to the webinar of today, the focus will be on neuroscience, so in the tissue topics. And we will start with Dr. Tommaso Fellini from the Italian Institute of Technology in Genova, that will talk about advancement in deep brain imaging. Then we will move to Scanless two-photon voltage imaging with Dr. Imane Bendifallah. And we will end with Dr. Francesco Gobbo from the University of Edinburgh, that will show us how we can use a miniscope to miniaturize microscope to disentangle computation in the hippocampus. So before starting, just a brief technical point. Questions are allowed at the end of the seminar, so from 4 p.m. on in the follow-up session. And there are two ways in which you can ask questions. First, during the entire session, you can write them using the QA button, and then we will read them in the follow-up question. On the other hand, if you want to ask another question during the follow-up session, you can raise your hand and we will enable your microscope. So now, without any further ado, I will stop the sharing and let the stage to Letizia that will introduce the first speaker.
Yes, thank you, Matteo, and good afternoon, everybody. It is a great pleasure to introduce the first speaker of today, Dr. Tommaso Fellini from the Instituto Italiano di Tecnologia in Genova. Dr. Fellini graduated in physics at the University of Padua, studying enzyme kinetics, and completed his PhD investigating voltage-gated calcium channel and the functional consequences of diseases linked to mutations. He then conducted postdoctoral research on neuroglia and communication, first in Italy and later at the University of Pennsylvania School of Medicine. He is now senior principal investigator at the IIT, head of the optical approaches to brain function laboratory and co-head of the neural coding laboratory. He is the recipient of major European grants, including the ERC Consolidator Grant, Neuropatterns, and the FIS3 Advanced Grant Optica, and the co-founder also of the startup Smart Micro Optics. Today, we have the pleasure to hear his talk about aberration-corrected microendoscopes for extended field of view deep brain imaging in benchtop and miniaturized two-photon microscope. And now, Tommaso, the stage is yours. Thanks. Presentation. So, the second speaker of today is Dr. Imane Bendifallah. Dr. Bendifallah is a research engineer in Valentina Emiliani's group at the Vision Institute in Paris. She earned her PhD in neuroscience from Sorbonne University, and now she works on developing and testing optical and optogenetic tools for two-photon voltage imaging and all optical connectivity experiments. And today she will be sharing part of this work with a particular focus on scanless two-photon voltage imaging. So Imane, the stage is yours.
Thank you. Hi, everyone. Can you hear me well?
Yes, we can.
Okay.
Yes.
Thanks. So thanks for the introduction. Thanks for inviting me here. So I'm Imane Bendifallah. I work in Valentina Emiliani's lab, and today I will talk to you about scanless two-photon voltage imaging. Okay. So Valentina's lab focuses on developing optical tools for manipulating the neuronal activity via optogenetics. So by photoactivating or photoinhibiting neurons, and for reading out the neuronal activity using tools such as calcium or voltage indicators. And so my role in the lab, as well as my other fellow biologists, is to test these tools on biological samples to characterize their performances and to demonstrate their usefulness. So this manipulation and readout of the neuronal activity was traditionally performed using electrophysiological approaches. But since then, photonics approaches have been developed, and so we are using them because they display many advantages. The first of all is that they are less invasive. They can also adapt to relevant spatiotemporal scales, and they can be combined with genetically targetable tools that allow to study a genetically defined population of neurons. So we mainly use two-photon excitation in the lab because it has several advantages that Tommaso actually already mentioned. So the first one is that we use redshifted wavelength, so they are less sensitive to scattering, so we can reach neurons deeper in the brain. But also, the optical sectioning that is inherent to two-photon excitation gives us a single-cell resolution, and so we don't excite cells above or below the focal plane. To study a neuronal population using two-photon optogenetics, what you need to be able to do is to photoactivate or photoinhibit multiple neurons simultaneously and with a very good temporal precision. Unfortunately, the conventional scanning approach does not allow to reach multiple scales in a millisecond time scale. And also, the conventional two-photon spot is too tiny to actually activate the whole cell membrane. But so people have actually successfully scanned this tiny spot around the cell to activate the cells. But this technique is not as efficient with very fast opsins, which give us the best temporal precision. And so an alternative approach would be to extend the spot size to cover the whole soma. And so the lab has developed this technique called computer-generated holography. It uses a spatial light modulator, which is a device that can modify the phase of the incoming beam to produce shapes in the focal plane. So you can choose the shape that you want. Here, for example, was reproduced a dendrite. You can also choose to draw spots. You can draw multiple spots, so you can access multiple scales simultaneously. And so in our case, what we want to do is to extend the size of the spot to cover the whole soma of the cell and to excite it more efficiently. So one example of the work that we perform in the lab using these tools is this proof of principle of high throughput in vivo synaptic connectivity mapping. And so in this study that was published last year, Iwan, Joe, sorry, and Dimitri here, they combined this approach, so two-photon holographic photostimulation, and they combined it with electrophysiology to map the connectivity of layer 2-3 in the mouse visual cortex. So to do this, Iwan expressed the opsin chrome in the visual cortex of a mouse, and she then patched a non-expressing cell in layer 2-3, and then together they probed via two-photon holographic photostimulation, they probed the putative presynaptic cells. So here you can see an example of the results that they could get. So you can see, for example, that the target 2, when photostimulated, did not elicit a response in the patched cell, whereas the cell 22 here almost always elicited a response. And this indicates that target 22 and the patched cells were probably connected. So this was a very nice study. And now the lab wants to take this study a step further. So first, by making sure that we are indeed activating always the cells that we are photostimulating, so that we can dissociate the false negatives from the true negatives. And also another thing that we want to do is to actually remove this electrode here and replace it with light as well. And so in both cases, one way to do this would be to use voltage imaging. So to use voltage indicators are fluorescent proteins that are sensitive to light, and they transduce the voltage changes directly into changes of fluorescence intensity. So the most used in two-photon are these ASAP type-like indicators. They're composed of a voltage sensing domain here that is located in the membrane of the neurons, and it's coupled with a circularly permutated fluorescent protein. And so when a depolarization or a hyperpolarization arises in the membrane, the VSD undergoes a conformational change that induces a further change of conformation from the fluorescent protein that changes its intensity. So this approach allows for direct measurements of voltage changes, and so we can record action potentials, but also postsynaptic events. But there are many challenges that are inherent to the technique. The first one is the fact that the indicator has to be localized in the membrane to be functional, and so this reduces the number of molecules that we can record from. And the second one is that is the kinetics of the events. In the case of action potentials, these are very fast events, and so it forces us to record the fluorescence at a corresponding rate. And finally, in the case of postsynaptic events, these can be of very small amplitudes, which makes it difficult to record the optical signal. So all of these makes us work with an extremely low photon budget. And so our reasoning to tackle this issue was that by increasing the dwell time of the laser on the cell, we would maximize our photon budget, and so we would maximize our SNR. So we chose to sculpt the light using the techniques that I previously showed you. And so we coupled this light sculpting setup with a camera detection that could allow us to record from multiple cells simultaneously. And so the work that I will show you here has been done in collaboration with Ruth Sims here. She's a very talented physicist in the lab. She designed the setup, and we designed and performed the experiments together. So our first step was first to check that these scanless approaches were indeed suitable for voltage imaging. So to do this, we used at the time the latest developed indicator, which was the Jedi 2P indicator. It's a green indicator, like most of the indicators available with two-photon excitation. And it's a negative going indicator, which means that when a depolarization arises, there's a decrease in its fluorescence intensity. So we expressed this Jedi 2P indicator into cultured cells that we patched, and then we induced some steps of depolarizations. And as you can see here on the video and on the optical trace, the fluorescence matched really nicely the steps. So we also validated this approach on real neurons. So for this, we used organotypic hippocampal slices from mice that were expressing Jedi 2P as well. And we saw that we were able to maintain our delta F over F even in these densely labeled samples, thanks to two-photon excitation. So we were very happy with this, but what we actually wanted to record was real neuronal activity. So what we did then was to patch these same cells and monitor their their spontaneous activity using electrophysiology. So this is the black trace that you can see here. And at the same time, we were recording the fluorescence, which is here the blue trace. And as you can see, both of these traces are very similar. We're able to detect single action potentials, but also these trains of action potentials. And in this zoom in, you can see that all of the spikes are actually resolved. Here are other examples of what we were able to record with different patterns of activity. We can also see here some hyperpolarizing events or single action potentials. So we were very happy with these traces. And so we set up to characterize the parameters that were necessary to image neuronal activity. So using patch clumped, we electrically triggered action potentials and we recorded them at different acquisition rates and laser powers. And here you can see the results. So at 500 Hertz or at 1 kilohertz. And what we found out was that in our conditions, 500 Hertz was actually enough to record and detect action potentials in single trials. And although, as you can see here, the delta F over F of the 500 Hertz recordings were always lower than the ones of the 1 kilohertz, the SNR, on the contrary, was always higher. We also showed that we could detect fast trains of action potentials at all acquisition rates tested, with sub-millisecond precision in terms of AP timing. Okay, so but coming back to our connectivity mapping experiments that I was talking to you about, what we want to do is remove this electrode and replace it with light. So because postsynaptic events can be very small, we wanted to test first if we could have any chance at recording such small depolarizations. So we patched the cells. We induced small steps of depolarizations from 0.5 to 2.5 millivolts. We repeated this protocol 50 times, and then we averaged the repeats. Here you have the example of a cell where you have the results after 5 repeats, after averaging 25 or 50. And what we see here is that the more we average the data, the more we could extract the signal from the noise. In this graph here that shows the SNR as a function of number of repeats, we were able to see that depolarization of 1.5 millivolts and above were detectable after 5 repeats, and that after averaging 25 times, we were also able to resolve 1 millivolts. So this was a step in the good direction. I also have to say that this data was acquired at 1 kilohertz, but we know now, as I showed you before, that by recording at 500 hertz, we could increase our SNR, so thus reduce the number of repeats. And it's also important to note that we use Jedi 2P for these experiments, and although it's a very efficient voltage indicator, it was not particularly designed for these types of experiments, and so maybe an indicator that would display higher sensitivity in this range of depolarization would be more suitable. So all of this characterization has been done on single cells, but as I mentioned before, we have a camera detection that allows us to record from multiple cells at the same time. And if we go back to our connectivity experiments, We also gain a lot of time if we could probe multiple cells at the same time. So we wanted to know if by adding more cells in our field of view, we would degrade or not our signal. What we did to test this was patch a cell, it's the number 10 here, electrically trigger spikes and record the fluorescence response. So this is the trace that we got when we were only recording from it. And then we gradually increased the number of spots in our field of view. And so here's the results of our cell when we added 3 more targets, and here is the results when we added 15 more. What we were able to see here is that whatever the number of targets in our field of view, our delta F over F and SNR was maintained, which means that we did not degrade our signal, and it also means that we can indeed record from multiple cells simultaneously. So our last step in this project was to test that we could record spontaneous activity in vivo on multiple cells. So to do this, we expressed Jedi2P in the cortex of mice, as you can see here, and we recorded the spontaneous activity of several cells. So in this example here that I showed you, we recorded from 4 cells. Here's the average projection of the data we got. And here are these corresponding traces. And you can see that the cells displayed several patterns like single spikes or trains of action potentials here that are zoomed in here. And so this particular example was taken at 50 micrometer below the brain surface. But we were actually able to record spontaneous activity all the way to 200 micrometers below the surface. And so finally, I also wanted to briefly talk about voltage imaging with another type of indicators, which are the rhodopsin-based indicators. In this case, instead of a VSD, so the voltage sensing domain is replaced by a rhodopsin, and it's basically the voltage sensitive part of these indicators. So these indicators are widely used under one-photon excitation because they are typically much brighter and more stable, but they were previously thought to be incompatible with two-photon excitation. However, in the lab, we had Chrissy here, who just could not understand and figure out why this could not be possible. So she developed a new indicator that she called Jarvis. It's also a green indicator, as most of them are. And she expressed it in the zebrafish, and she was able to record spontaneous activity in the brain of the fish under Scanless excitation. So after this, she then tried other indicators that were previously published, like Voltron 2, and she also realized that they were as well voltage sensitive under two-photon excitation. So this one is particularly interesting because it's actually redshifted, and I will talk briefly about it after. So now that we are able to record action potentials, trains of action potentials, and potentially subthreshold events, what we want to do in the lab is to integrate all of these tools in all optical connectivity mapping experiments. So we are currently working on 2 different axes. The first one is to detect postsynaptic potentials, and the second one is to combine voltage imaging with optogenetics. So for the first one, as I was saying before, the tricky thing is that unitary postsynaptic potentials can have a very small amplitude that can go actually even below 1 millivolt. And so the ideal voltage indicators to record this would show a higher sensitivity in these ranges of depolarization and a high SNR. Here, as a reminder, is the data that we had gotten before with the Jedi2P. And we were able to resolve 1 millivolt after 25 repeats. So we have been testing many different indicators from our collaborators. Here is one of the last ones that we tested. So this data is very preliminary, but here we showed that after 25 repeats, we were actually able to resolve 0.5 millivolts. We think we could go even lower, but we still haven't done the experiments. So we will have to see, but this is very, very promising. And so for the second part, which is combined voltage imaging with optogenetics, as I was saying, most voltage indicators that are efficient in two-photon are functional in the green range of the spectrum. But unfortunately, also, so are the opsins, most of the opsins that are available. And actually, even the red shifted ones, like the crimson R, still display a non-negligible activation in the blue shifted wavelength that we would use for voltage imaging. And so we have spent a lot of time testing different combinations of opsin, of indicators, trying fast or slower opsins as well. But we could not manage to get a crosstalk-free combination where we would not activate the opsin when imaging. And after a while, we realized that this could actually be somehow super useful for our connectivity mapping, because as I was saying to you at the beginning, we also want to know exactly what we are doing to the presynaptic cells. So we know for now that we are photostimulating, but we never know for 100% if the cell has spiked or not. And so actually, we could take advantage of this huge crosstalk that we have here and use the single beam that we use for photostimulating, just add a voltage indicator to these cells and record the response from the cell. So we tested this on organotypic hippocampal slices. We expressed both Jedi2P and the chrome, and you can see here, so the chrome is the green curve. We can see here that it's very much activable at 940 nanometers, which is what we are using for Jedi2P. For these experiments, we patched the cells to monitor what was happening when we would shine the light. And so these are the results, and here is a zoom in. So what you can see is that as soon as we shine the light to photostimulate, we get an action potential. But since we are also recording, we realized that we could also detect the spikes. So this could help us to dissociate from the false negatives and true negatives in our connectivity mapping experiments. But there are also many, many other experiments that we could do. And in this case, we would not want to have this crosstalk. So we would want to be able to record the voltage of many cells. And when we want it only, to photostimulate by photostimulating with another wavelength. And so this is where the Voltron 2 comes back. As I was telling you, this one is activable at 1100 nanometers, so we can record with really red shifted wavelength. And we know that there are also many opsins who are more active in blue shifted wavelengths, like the PACHR here, which is the dark blue curve here. We have shown previously that these opsins can be activable at 850 nanometers. And since this opsin is not active at 1100 nanometers, we have hope that we could actually combine both of them and really get a crosstalk-free combination to perform crosstalk-free experiments. And so on this note, I want to thank first Valentina Emiliani, of course, the rest of the voltage group in the lab, which is Irini, Ruth, and Chrissy, the whole lab, of course, our collaborators, and again, you. So thank you.
Thanks a lot, Imane.
Okay. Thanks a lot for your interesting presentation. So now the stage is for our last speaker, which is Francesco Gobbo. If you can...
Yeah, sure.
Okay. So, okay.
So good afternoon, everyone,
Oh.
and thanks for having me. It's really a pleasure.
Yes.
Please interrupt me anytime if you want. But essentially, my talk is on more the application side of the techniques that Tommaso and have presented. So in terms of using like miniscopes to acquire the topics that I'm interested in, which essentially is memory. So what is memory? I mean, we have a very intuitive concept of memory as of remembering things, remembering events. So essentially having in our brain a storage of past events and past information, and to use that to navigate our own life. It is such an important part of our life that sometimes it's very easy to forget how much of our life is actually due to memory, and take it for granted. So it might be easy to appreciate how important it is when our memory actually fails us. So, for example, in various forms of dementia, like for example, Alzheimer's disease, frontotemporal dementia, and so on, actually one of the first symptoms is memory loss. And such is the power of memory has in guiding and impacting our life. Not only neurodegeneration, like in terms of Alzheimer's, but also other forms of neurodegeneration, like for example, the famous example of Cleo Waring was a famous musician and composer. So he suffered from severe deficit in forming new memory and accessing past memories due to a very aggressive form of viral encephalitis. And what happened to Clive is that basically he was stuck in a constant present. And this is just an excerpt of his diary. And you can this is like a page of thousands of those. And you can see that at every minute he was, as waking up from a past experience, but without necessarily knowing what happened to him and what's going to happen to himself. So such is the impact that the memory has in our life. So in terms of memory, then, a memory can be defined as the act of remembering. And usually this is accompanied by the knowledge of that being a memory. So we experience like a past experience, but we are aware of that not being the present that we are currently living in. And we use this acquired knowledge to change the response to novel stimuli. And in that sense, it has an clear evolutionary advantage in terms of shaping our behavior and being able to respond to novel experiences. In terms of memory processes, what we normally distinguish is between a phase where we acquire a new type of information, which is generally defined as encoding, how it actually represented in the brain, so where it is stored and which form it is stored. But because we keep some, but not all of the information that we acquire on a daily basis, what is that consolidated and expressed at a later time in our life. So the focus of my talk today is talking about spatial memory, which, as Tommaso already introduced, is memories about space and the location where we are, but also remembering episodic memory, so essentially remembering events that have occurred in particular spatial locations and contexts. So from a very didactical point of view, there are other forms of memory, for example, working memory, which is related to storing and having access to temporary information, like for example, would be tapping a phone number that somebody has just told us shortly before. Procedural memory, which has to do a lot with motor memory and with the motor areas of our brain, and for example, could be how to play the piano or even digiting the numbers on our phone without looking at it. In terms of long-term memory, we have like a major distinguish between episodic memory. So essentially, in first approximation, memories of facts that happen to ourselves. Spatial memory, or otherwise memory for locations, and semantic memory. Semantic memory is kind of a particular form of memory that abstracts from single events in our life, like episodic spatial information, and kind of sublimates, like for example, we have a general knowledge of a horse as opposed to, I don't know, remembering when you had like a particular pony when you were growing up. Both spatial navigation and episodic memory are very much linked, and often they are related to sides of similar aspects in humans as well as in mammals. and the brain area that's devoted to the processing of spatial information and episodic memory is in first instance the medial temporal lobe. So the medial temporal lobe is composed of the hippocampus proper and other cortical areas associated to it, like for example the peroneal cortex, the medial lateral internal cortex, and the dendri gyrus. How can we can access memory from a neurological point of view? So a particular phenomenon that's been described early in the 2000s was the discovery of replay. So what the authors find, from the Bijaki group in particular, was that when recording from the hippocampus of mice and rodents in general running through a linear track, was that as these animals navigated through this linear track, a sequence of cells responding to the position of their environment would fire in sequential order, of course. And what they found is that in moments of quiet, before or after the running again this in this linear track, they would find like a accelerated reactivation of these cells in either forward order, so maintaining the order where these cells were actually active during the linear track, or sometimes in reverse order. So replay has been mostly described as a mechanism that was thought to support memory consolidation and the essentially storage of new memories from the hippocampus with the interplay of other brain areas. But recent evidence has suggested that replay is also a possible candidate for memory access. And this is because when recording from animals that are about to solve a task or remember a particular location, often replay is recorded from the hippocampus. However, the situation is much more complicated than that. So different experiments with different behavioral paradigms have generated very conflicting results. So for instance, some experiments have shown that the replay that can be recorded from the hippocampus before animals make their choice mainly reflect the previous location where the animal has been to, whereas other experiments has otherwise linked it to future locations. So has been suggested that a replay has been used to guide a decision about where the animal is about to go. Some other experiments have shown that also remote or even avoided space can be replayed, but for example, the animal is faced with the possibility of taking a turn to an area where, for example, he received a food shock, so essentially where they want to avoid entering again, and instead they do, researchers do find a replay of cells corresponding to the allocation. And further than that, also the replay of complex or even impossible routes have been recorded. This is an experiment where the animal was free to move around in a two-dimension with barriers that were moving around so essentially preventing the animals from taking some transitioning, say, for example, from one corner to the other because there was a barrier in between. And when looking at the replay, they would actually find sequences corresponding to the navigation, the theoretical navigation from one point to the other, passing through magically through these barriers. So one, from a theoretical point of view, one possible solution has to do with the role of the hippocampus as possibly being used as a way of constructing possible hypothetical scenarios. And this resonates with data from the human patients that suffered hippocampal damage. And this is the work that Eleanor Maguire really pioneered. So what she found is that whereas in healthy controls, people were able to form like very complex and detail-rich imagination and description of scenes as request, patients with hippocampal damage were only able to provide very general and very superficial description, and actually the majority of the information that they were adding to their stories or their description were of largely a non-visual nature. Like, for example, when patients were asked to describe the imagining being on a beach, they would only provide information about the color of blueness, or, for example, the sense of warmth or the sun under their feet, but not much more. Whereas healthy controls could provide like a full scenery, like more or less what's being depicted in the picture. So besides processing memories, the hippocampus is also involved in the representational space. So this has been now very much gained support from both loss of function experiments, so wherefore when patients and animal models, when suffering damage to the hippocampus, they have a severe impairment in navigation, but also sometimes gain of function experiments, like, for example, and this is again work by Eleanor McGuire, showing that taxi drivers in London, that in their processing of acquiring their taxi license, they memorize essentially all of the routes and streets that make up central London. So what she found out is that in the population of taxi drivers, as opposed to other drivers that didn't really memorize the whole network of roads in London, the volume of the dorsal hippocampus in particular was significantly higher than healthy controls, than I would say normal controls. So how do we actually study spatial memory in the lab? So essentially we use a lot of mazes. One of the most paradigmatic mazes is the Morris-Water maze. So essentially what we do is a number of learning trials where animals learn, for example, the position of a platform in escaping from a two-dimensional water arena, and then essentially they are tested if they remember the position with the platform not being accessible or even visible to them. So what has been found is that this task is dependent on the activity of the hippocampus, and it also has been widespreadly used in linking new learning with synaptic plasticity. Just a second. So it's just loading the next slide.
It's not anymore in presentation mode. I don't know if you... Okay, yes.
Oh, is it now?
Yeah.
Oh, sorry.
No problem.
So apologies for posting this again, but this is the same video that Tommaso showed you earlier. So this video by Roddy Graves, who actually was at University of Edinburgh before joining K. Jeffrey Lab in London and now at Northwestern. So essentially, researchers over the years and decades have found the supposed neural correlates of space representation in the brain. So this, as Tommaso was showing you, is the recording of place cells. So as animals and also human evidence have shown, in the hippocampus, we have like cells responding to the position of the subject, and therefore providing a so-called cognitive map, as John O'Keefe described it, representing the position of space. So beyond place cells, I mean, a wide variety of spatial selective cells has been described in the brain. For example, data from Edvard and May-Britt Moses has defined grid cells in the internal cortex, head direction cells in the anterior thalamus and other brain areas. So overall, a number of groups have described a family of spatially selective cells that essentially allow the representation of information either in allocentric or in egocentric ways. And I will explain in a second what allocentric and egocentric means. But essentially, the interplay of these systems of cells is believed to overall allow the animal or the subject in general to reconstruct from the environment where its position in space is. So the distinction between egocentric and allocentric is essentially how an object or a location is represented with respect to the coordinates in the environment. So in the egocentric case, for example, you define objects with respect to your own position. So for example, you remember that, I don't know, a library or a bicycle is to your right rather than to your left. Whereas in allocentric way, you define object with respect to their own position. And this is very much similar to what we use, to our experience when we use, for example, a map to navigate like a novel city. So not only humans, but also rats can be trained to solve a task in general using either allocentric or egocentric strategies. So the question in general that we were interested in is how the spatial information that we have in the brain, or rodents have in the brain in this case, is modulated by the task, in this case by the strategy that animals use to solve a particular task. And second, how is the spatial information used to retrieve memory and guide decision making? So to answer this, we made use of miniaturized miniscopes or miniscopes. So, well, Tommaso did a great deal of wonderful explaining how essentially green lenses and miniscope work. But to just briefly recapitulate, miniscopes are essentially contain the minimal elements of microscopes and allow the recording from deeper brain structures of the activity of neurons expressing a fluorescent sensors. In this case, we were using GCAM6Fs. So this is the semi-process typical recording that we obtain when animals, and in particular rats, are moving around. So rats are free to move in a very wide environment. The arena that we're using is larger than 2 meters. So they have the possibility to explore this environment, retrieve food pellets at different locations, and then essentially test them for their memory. And at the same time, we can record what happens in their hippocampus by looking at these flashes of neurons in essentially in CA1. So the advantage of using miniscopes compared, for example, to electrophysiology is to be able to follow really the same neurons from day to day. And so this was allowing us to ideally solve some of the issues that the literature found when describing the role of replay. And the reason is because by means of electrophysiology, people and researchers in general typically recall from trial after trial with the position of the target changing between consecutive or closely spaced trials. Therefore, the confusion between what's past and future target really is hard to disentangle in some experiments. Whereas in our, we design an experiment so that animals would retrieve rewards and food pellets in a different position every day. So therefore being really able to tell apart whether that was an experience of the past or a future action by the animal. So the task that we use is based on the so-called event arena task that was developed in the laboratory of Richard Morris. In principle, it's very similar to the concept of the Morris water maze, but with the main difference of being performed on land rather than in water. So animals start each session from one of possible different locations called star boxes, and so which are essentially just small enclosures where they start their trials before entering the two-dimensional arena. And in the arena, they can get food pellets from one of 6 possible sandwells. So essentially, the food pellets are stored within a small container on the floor, which are called sandwells. And on a given day, only one of those is rewarded. After 1 hour to 90 minutes, we test whether animals have learned which one is the rewarded sandwells. And if they are correct, they would essentially visit the rewarded sandwell first. And so a session over session, then we change the position of the rewarded well by randomly selecting one of the 6 of those, and that goes on for several days. On this overall structure, we are able to have animals use either egocentric or allocentric strategies by modulating how they solve the task and which starting points we're using for each session. For animals to use an egocentric strategy, essentially for each session, we have animals starting always from the same starting point, or start box in this case, and essentially they go back and forth from the starting point to the rewarded well over and over a number of trials in the same session. While to model allocentric strategy, animals are required to reach the same location starting from different starting points in the same session, and also not coming back to the original point to consume the pellets they have retrieved, but actually go back to a fourth position, which is essentially called gold box, to essentially be able to eat the pellet. So we have therefore trained the animals for a number of sessions. So we have two groups of animals trained either in the allocentric or the egocentric protocol, and then they are counterbalanced and flipped to the other protocol. So we do run a number of tests to make sure that they are really using egocentric or allocentric strategies. For example, allocentric animals that are performing the task allocentrically have to rely on the position of available cues and hallmarks in the environment. If we remove those and we mask the extra maze environments with curtains, for example, animals are not able to orient themselves and essentially they start searching the rewarded well at chance, while egocentric animals are really much unaffected, so they can retrieve the rewarded well just as easily as with cues available. And then we tested, for example, in a separate number of tests, we test if egocentric animals are actually using the cues or not. So in particular trials, we test the animals from the opposite starting points and with the opposite rewarded well, as you can see, for example, in panel G. And so essentially you have a conflict between allocentric cues around the position, but the animals are able to retrieve the sandwells, which is correct in egocentric terms, even though it is incorrect in allocentric terms. So we then went on to look at what the activity in the hippocampus. So the first thing we checked is whether we had place cells in allocentric animals as expected, and in egocentric animals. So this was a bit contrary to what we were expecting. So we were expecting like bigger differences between the two groups. But what we found is that indeed allocentric animals had more cells that would be classified as place cells compared to the egocentric group. We thought that this was largely due because in the egocentric group, we found more cells that had more than one place field. And this is probably the reason why the number of cells being classified as place cells is lower. And it's because we use, as Tommaso was briefly saying, in defining place the place cells by means not of mutual information between the neural vector and the position vector of the animal in the arena. We also try to compare the representation of symmetrical trajectories, and this is really where it went completely off track compared to what we were expecting, meaning that we were, in our mind, we thought that if animals were performing egocentrically symmetrical trajectories and were not using the allocentric cues available to them, the representation of these trajectories will be somewhat similar to each other, because at the end of the day, if in their mind they were repeating the same trajectory, there was no reason why they would be dissimilar at the neural level. The reality was the total opposite of that. So while we did find that allocentric animals did have a higher structure in their trajectory, nevertheless, I mean, the representation in terms of neurons active between the two trajectory in the egocentric animals was still different, and we could still distinguish the two trajectory by looking at the activity of neurons in between the two of them. So this suggested us that the hippocampus still represent, in a sense of passive way, the position of the animal just based on the available cues to the subject. We next went on to answer our second question, so how is spatial information used to retrieve memories? That was based on pilot experiments that were published previously. So what we have found is that when the animal is in the star box and you see it's like standing still, it's not that there's no activity, and actually we can decode where the animal is about to go. So for example, here you'll see that in the star box there's some activity, and over time we can see that our algorithm predicts like different destinations, if you want, and then eventually what we find out is that the animals at some point takes a decision, exits from the star box, and actually gets to the correct sunwell. So what we did find is that we were again able to predict where the animal was about to go, but only where the animal was making a correct choice. And instead, that wasn't the case where the animal, instead of going to the rewarded wells, was making a mistake, so going to a sandwell which wasn't didn't contain any food pellets. So, and this was, and again, what we found is that while the animal was in the star box, there was reactivation of cells that corresponded to potentially different trajectories and destinations. And this is, for example, different cells that were active while the animal was in the star box, and this is what they represent in terms of space. So you see that, for example, 5 seconds before leaving the star box, the animal's thinking about, well, OA cell is active, that is representing the destination, to the sunwell is represented. At -3, the analyst start thinking about a different one, and then again focuses back on the rewarded one. So we did try and extend these results to a bigger arena and the allocentric and egocentric trained animals, and indeed we find again that there was a much higher than chance reactivation of cells representing sunwell's location while the animal was in the star box about to start their trial. And this was also confirmed with a detectors where sunwells were much, the decoding of class of sunwells was much higher than chance and much higher than other points in the arena, still considering the star box activity. But we found a difference, meaning that allocentric animals, as you can also see from the number of the fields of the cells which are active in the figure, is that while allocentric animals were thinking about different possibilities, but at a given time the cells active were representing quite robustly and quite specifically a specific sunwell at a given time points. What we find in the egocentric case is that what was reactivated was something with a lower spatial resolution. So, for example, at some point you would find cells that were mapping more than one sunwell, or even without having any obvious correlation to sunwells specifically, and rather just being active at random position in the arena. So we then went on to see how this reactivation is linked to future decision, and indeed, again, we found that in the star box we had a higher reactivation of cells corresponding to the correct sunwells, but that was only for in trials where the decision was correct, and not in, again, trials where the animals made a mistake and went on to the wrong sunwells. So we then asked whether the animals were instead thinking of the first visited sunwells, and indeed we found that there was like a significant trend towards that, meaning that when animals were making mistakes, it's not that they were thinking of nothing, but rather they were thinking to a higher chance of the actual position where they would end up being. So, for example, And this was before any previous knowledge of the location of the rewarded well. So this, for example, is an example from the first learning trial. So the animal has no experience where in that session the rewarded well would be, and still we find like comparable reactivation of sand wells even in this absence of prior knowledge. So the animals of course have knowledge about the arena, but not about which is the rewarded well, and we still find like activation of the possible sand wells. However, this is not that simple, so it's not as clear-cut. So the animals are only thinking about the destination where they're about to be, or in other ways, the correct sand wells. So what we found instead is that a lot of possibilities are played out by the hippocampus, with possibly like a focusing towards the decision-making point, towards the actual future sand well. So overall, our data suggests that the hippocampus is not only predicting where the animal is about to go in definitive terms, but rather is playing like different possibilities as if the animals are making like mental time travels, which is again very similar to our concept of memories, and see what would happen if they would end up in different locations. So again, then we find in the star box, so during decision making, multiple destination can be represented from the same starting point, and again, vice versa, the same population of cells could represent the same destination when starting from different starting points. We then asked if this activity that we were measuring by means of miniscopes was necessary to actually navigate to the correct sand well. So to do that, we deemed to inactivate essentially the hippocampal activity in this time window where animals are making that decision. So to do that, we express a optogenetic inhibitory channel, which essentially is JAWS responsive to red light, or as control, we just express GFP in the hippocampus of a separate groups of rats. In control animals, we found that we were able to effectively silence the activity of neurons in CA1 quite robustly, and also reduce the number of ripobinds, which in general are associated with memory recall and replay events. So that's what we found. So this is an animal where we didn't shine the light. So this is a typical example. The animal is in the star box, and then at some point it just gets out, remembers what it should be doing, and gets the pellet, and that's it. And so this is what happens instead when we are shining light into the hippocampus during, again, this decision-making time point. And so the first thing you notice that the animal is spending longer time in the star box, as if it's a bit more confused about where to go and doesn't really know what to do about it. And then we'll see what happens when he gets out. So actually the first thing he does is make it a mistake. And then basically you will see that he commits a number of errors before actually getting to, this is actually the same animal as before. So essentially, eventually he will get the correct sand well essentially by chance. So what we found then is that the optogenetic inactivation of hippocampal neurons impairs the performance in animals that are solving the task using allocentric strategies. But strikingly, animals that were using egocentric strategies were pretty much unaffected by it. And when we also inhibited not only the decision-making time point in the star box, but the whole trial in the arena, the performance dropped even further, demonstrating that the allocentric task is dependent on the hippocampal activity, while the egocentric task is not. So in summary, Our data support a role of the hippocampal to, well, construct potential alternatives by essentially playing out different possibilities. And this is what we believe in terms of can link spatial navigation and episodic memories, because a lot of episodic memories and a lot of memories in general are not just, well, flashbulb reactivation of experience that we had, but they always have a reconstruction component to them, meaning that we know what we probably remember is the gist of the memory, and then the hippocampus actually construct a scene with a richness of details. So in this sense, the hippocampus can be regarded as a brain area that has the ability of bridging past and future. So we recall past events and we access the information about to make useful about the present, and can also be used to imagine future and different possibilities. And last, I would like to, well, thank everybody that was involved in the project. In particular, Rufus was a PhD student that was working with me and did a lot of the coding. Richard and Simon for supervising, and Mark for help with the miniscopes. And then again, all of my colleagues and students that participated to the project. And of course, thank you for listening.
Okay, thanks a lot, Francesco, for this fascinating presentation. So now it's time for the question. So if you want to ask another question, please feel free to raise your hand. Let's see if we... Okay. So no question at the moment. So I will start. I have a question for Tommaso about the corrective lens that you are placing above the gradient index 1. Are there some constraint or issue in aligning everything, or it's pretty straightforward?
So aligning is obviously fundamental to have optimal performance. I tell you that we do optical alignment under visual inspection under the stereoscope, and this, I mean, is a delicate but not too difficult of a process. So different people do it very effectively.
Okay, thanks.
And to be, yeah, maybe I can add this. It is facilitated by the fact that the corrective optical element is attached, is printed attached to the cover slip. So you're handling the cover slip and you're aligning basically the corrective optical element holding the cover slip, which is a bit bigger than the corrective optical element. And so that is easier.
Thanks.
I have another question for Tommaso. We started with you. So I was wondering whether the movies that you showed were acquired while the animal was free to move, or they were had restrained. And if they were free to move, which strategies are you using to register the movies?
So then the movies in which you had the mouse moving on the left and the calcium imaging on the right, they were acquired in 3D moving animals. So and they are